So hi, today we are going to discuss the first lesson. Actually, it is not a first lesson. The first lesson is the cell division. So this is the second lesson of the pathology, that is the important lesson, that is cell injury, cell death and adaptations. So today let's discuss this lesson in a single video. So here I have shown an image. Actually, these are the notes which I have created myself. So let's discuss this lesson in a simpler way using this note. So the first here, the reference which I took is only from Robbins. So this concise note is completely or 100% from Robbins. I took this. So let's start. So first, normally a normal cell, if it undergoes stress or if there is an injurious stimuli, they would undergo adaptations. For example, if a normal cell undergoes this, they would undergo adaptations. If the cell is not capable to adapt or if inability to adapt, what happened? Then there would be a cell injury. So cell injury may be directly caused by an injury stimuli also, but it may also due to the inability to adapt. And injury is like it, if it is reversible, then the cell can again go to its normal state. It can go back to its normal state. If it not, then the cell would undergo the irreversible injury. If the cell injury is irreversible, then definitely they would undergo death by either of this necrosis or apoptosis. Okay, so first let's see what are the causes of this cell injury, how the cell get injury. So these are the major causes, and here the most important and the primary cause is hypoxia. So hypoxia is nothing but, of course, we all know that it is nothing but the reduction supply of oxygen, or we can tell if the oxygen supply is uh, altered or if it is reduced, then what happens to the cell? Then the cell get injured. And then the second cause is physical agents, which is due to any mechanical trauma or due to any increase in temperature or maybe due to an electric shock. Then chemical agents, of course, it may be due to the abnormal or we can tell the altered electrolyte, and maybe also due to the trace amount of poison. And then the chemical agents, which is, yeah, as I said, chemical. Then infectious agents may be due to any like virus, bacteria, or any other infectious agents. And immunological reaction, even immunological reactions, we can know that like an autoimmune. In those type of diseases like autoimmune disorders, the cell, even to the self antigen, the cell may uh, react in a different way. Okay, so that remain exaggerated reaction or injurious reactions to the self antigens. Then genetic abnormalities like the congenital genetic disorders, then nutritional imbalance. So all those things are the causes of cell injury. Now next, let's see the pathways of cellular response to stress and injury. So how the cell responds to the stress and injury. So first, integrated stress response, and we have three pathways. The first one is integrated stress response. So here what happens is, in the, okay, there is an stress sensing kinases. So the examples are protein kinase, then that is PKR, protein kinase R, then PKR like ER kinase, then general control non derepressible 2 kinase is GCN2, and regulated, it is heme regulated EIF2 alpha kinase, that is HR1. All these stress sensing kinase, what they do is they would sense the stress, and then they would pause. If the stress is sensed, then what happened? They would phosphorylate the EIF2 alpha, because all these are kinases, so they can phosphorylate. So EIF2 is an eukaryotic translation initiation factor. So when this factor is phosphorylated, they mediate two key response. That is global repression of protein. That means they would repress the protein synthesis, and also they would activate the transcription factor. So that control gene expression which promotes cell repair and survival. Okay, so by these two key responses they respond to the stress. So here, as I said, we see the protein a pkr like kinase, that is perk. So they would activate the unfolded protein response pathway, whereas the other kinases would activate autophagy. Okay, so autophagy we will see in the next page. So first let's see about the unfolded protein response and ER stress. What this means is, normally in our endoplasmic reticulum we have an uh, protein called chaperones. So chaperones, what they do, they control the folding of proteins, and also if any misfolded protein is found, what happens is they would proteolized in proteosomes. That means the chaperones would control the proper folding of protein, and also the elimination of misfolded proteins by proteosomes. But if the protein folding demand is increased, so the endoplasmic reticulum have a capacity, certain capacity for protein folding. If the demand increases, what happens? Then they cannot able to tolerate that. So what happened? There will be an accumulation of unfolded or misfolded proteins. So if there is an accumulation of these proteins, what happens is this unfolded protein response pathway is activated, and they would cause the increased production of chaperones, and also they cause the degradation of these abnormal proteins. So by, and also there would be a slow protein translation. This is what UPR. So we will call also this is endoplasmic reticulum stress. Why? Because of this accumulation, the endoplasmic reticulum undergoes stress. So they activate the UPR. Then the UPR would increase the production of chaperones, and they increase the degradation of protein, and also they slow the protein translation to prevent the further accumulation. But if this UPR fails, then the cell would undergo apoptosis. So the UPR is nothing but it is in response to the endoplasmic stress, but if it fails, then the cell can undergo apoptosis. So now we have seen that, okay, if a protein misfolding happened, then all this, then what are the causes of this protein misfolding? That means mutation. Because mutation, what happens? If a mutation occurs, then there would be an, so if a DNA is damaged, then there will be no formation of proper RNA, then no formation of proper protein. Even aging, plasma cell tumor, viral infection, then increased demand of insulin, because insulin is also a protein. If the demand is increased, then change in intracellular pH, then deprivation of glucose and oxygen. So all this, all these things would cause protein misfolding. Okay, and the next is the third responder is autophagy. So the first is integrated stress response. They indicate that how, what are the stress sensing kinases and how they activate the pathway, and what are the pathways activated. And in that we have seen UPR, and another is autophagy. So autophagy is nothing but it is a survival mechanism during nutrient deprivation. That means, for example, if due to some any other condition in for example, in some state, maybe what happened is sudden endemic or a drought or famine, it may lead to the scarcity of food, all those things. In those times, what happened? Suddenly, there would be a nutrient deprivation. So then the people can't, they didn't eat properly. Then there'd be nutrient deprivation. So by the time the what the cell do is they eat its own cell content for survival mechanism. Because if it doesn't get nutrient externally, what happens? They would eat its own cell content to survive. That is what autophagy. So here if we can see an simplified flowchart for this. So if there is a nutrient deprivation, there will be activation of initiation complex. So after nucleate the initiation membrane, sorry, not after ATG. ATGs nucleate this initiation. Nucleate means they, the what? Yeah, the ATG. What is ATG? Is nothing but the autophage triggering gene. So when these ATGs nucleate initiation membrane to form phagophore. So there would be an covalent linkage of lipid phosphatidylethionamide to LC3. They both get link covalent linkage. Then there will be an elongation and closure of incision membrane by capturing the cytosolic cargo to form autophagosome. So then the autophagosome fused with lysosome to form autophagolysosome, because we know that lysosome contain the enzymes for the hydrolyzed enzyme. Then all those content are degraded by lysosome enzymes. Then again the metabolite can be recycled. So by that the cell can able to survive. So temporarily it can able to survive by eating its own content. The next, what are the uses for this autophagy? Where it is used mainly to clear intracellular debris, for nutrient depleted cells, and also for turnover of organelles. Then let's see what if autophagy is dysregulated. If there is no proper regulation of autophagy, then they can lead to cancer, the neurodegenerative disorders, infectious diseases, and inflammatory bowel diseases. Then, so now we have seen the pathways, okay, pathways to the stress, how the cell acts. Now let's see the reversible cell injury. So what is reversible cell injury? As I already said, if a cell is if there is a reversible cell injury, then the cell can go back to its normal state if the what the triggering factor is removed. Okay, so the cell injury causing factor, if it is removed, then reversible can injury can like if a cell is reversibly injured, it can go back to its normal state by removing its triggering factor for the injury. So first let's see reversible cell injury. The generalized swelling of cell and its organelle, flibbing of cell plasma membrane, detachment of ribosome, and clumping of nuclear chromatin. All these, if there is a finding or like this, then we can tell of course that there is a reversible injury. So the cell can able to go back to its normal state by removing the triggering factor. Because many MCQs they can ask that what are the below options shows reversible injury. Even the NEET PG question they may ask like that, which of the following option is a reversible injury, or which of the following option is not a reversible cell injury, like that. So generalized swelling, blibbing, detachment of ribosome, and clumping of nuclear chromatin. We can see if there's an oxygen deficiency, as is it hypoxia, then decreased oxidative phosphorylation, then failure of ATP. Of course, we know that sodium potassium pump they function only by this ATP dependent pump. So there's a failure of the function of this pump. Then of course there would be an abnormal accumulation of electrolyte. Then there will be an inflection of water. Then cell gets swelled. So this is till now this is reversible. Okay. So if, now if the oxygen supply is restored, what happens? Of course the cell can go back to its normal state. Then fatty change. Fatty change is nothing but mainly it occurs in liver, because we know that the metabolism, fatty metabolism is mainly dealt by liver. So when the toxic injury, okay, what happens? There would be an accumulation. So accumulation of ATP. So fatty change is also an the main what we can tell. Sorry, it is also in one of the reversible cell injury like that of hypoxia or oxygen deficiency. So in a toxic injury, when there is a toxic injury, what happens is there would be an disruption, or we can tell there would be an disrupt of the metabolic pathway. When a toxic injury is there, what happened? The metabolic pathway get altered or disturbed. So there would be an accumulation of TAG. Now, third one is dilation of endoplasmic reticulum. It may cause detachment of ribosomes. Then, in mitochondria, swelling, appearance of small amorphous densities, and accumulation of myelin triggers in cytoplasm, then nuclear alteration. That is this segregation of granules and fiber elements. All these are the reversible cell injury. If the cell injury is irreversible, then the will undergo death. That is cell death. Now let's see about the cell death. We have two main pathways by which the cell undergo death. One is necrosis and another is apoptosis. So necrosis. So necrosis is always pathological. Apoptosis can be physiological or pathological, but necrosis is always pathological. So let's see the causes of necrosis. So the causes are ischemia, exposure to microbial toxins, burns and physical and chemical injury, active proteases leak from cell. So these are the causes of necrosis. So let's see the simplified flowchart for the necrosis. So say these are the causes. For example, um, if there is an some physical injury, or if the cell membrane is damaged, if the damage is severe, that means it is irreversible damage. Damage is severe. What happens is the lysosomal enzyme inside that cell, they would digest that cell. And of course, what happens? The cellular content would leak through the damaged membrane because already the membrane is damaged. So what happened? All those content would leak into the extracellular space. Okay. Then that substance is released. No, so they are called as DAM. That is damage associated molecular patterns. And this substance is not. It should not always might not be abnormal substance. Even the normal substances can be a dam. For example, even the ATP, even the uric acid, it is a normal substance which is present in the cell. But when it is released into the extracellular fluid because of this um, what the leakage, then they can be called as damp. Okay, so uh, this is this dam is played as an important biomarker. Okay, for example, as I said, a cardiac muscle cell, if it is injured, and what happened, the substances inside that muscle would release. So what are the dam which may be released means, for example, troponin. So if a cardiac muscle cell and the cellular content leak from that cardiac muscle cell, then the substance which may be released would be troponin. So troponin is here the dam. So if a troponin is identified in ECF, then we can tell that there is a cardiac problem, there is an injury to the cardiac muscle. Okay, so we know that troponin is one of the main biomarker in cardiovascular diseases, right? And also even ALT is a biomarker in mainly in the case of liver or gallbladder related disorder, because the ALT can be released if there is an injury to the bile duct epithelium. Even if a liver cell, the hepatocyte is injured, mainly the AST and ALT can be released. That is, yeah. So all those are also damp. So these are act as a biomarkers. So by which we can identify which cell get injured or the origin of injury. Okay, now let's go back to the flowchart. Okay, so the release of the dam, then dam would recognized by the receptors of macrophages and other leukocytes. Of course, what They do they trigger the phagocytosis of the debris and also they would product cytokines. What cytokine means? What? Yeah, cytokine causes inflammation. Then the inflammate after the inflammatory cell, right? So they produce proteolytic enzymes, and the proteolytic enzymes cause enzymatic digestion. So this is how the necrosis occurs. So in the necrosis, there would be a damage to the cell membrane and the leakage of cellular. That is very important feature which differ the necrosis from apoptosis. Even inflammation, the word inflammation you wouldn't see in the apoptosis. So that's the important feature which differ the necrosis from apoptosis. Now let's see the next one. Now let's see the morphology, how the cell appears. Okay, how the morphology when we see the necrotic cell through the microscope, what we can see. What is the morphology is nothing but the necrotic cell would be eosinophilia in appearance. That is, you can see like a pinkish. Why eosinophilia? Is because the cytoplasmic proteins which denature, no? So the accumulation of those cytoplasmic proteins, they would bind to the eosin dye. There is a red dye. So that way there is eosinophilia. And also we can see glassy homogeneous appearance because due to the loss of glycogen. Then what are the nucleic changes? The three changes in necrotic cell. The three primary or key features are karyolysis, pycnosis, karyorhexis. Karyolysis is nothing but basophilia of chromatin fade. Okay, and also the pycnosis is nuclear shrinkage. Karyolysis is that pycnotic cell would undergo fragmentation. Okay, so and then the necrosis we have the different types of necrosis like coagulative, liquefactive, caseous, fat necrosis, and all these. We have many. So here in the note actually I didn't write all those types, but I can discuss the types in a brief way, in a short. Okay, so first let's see the coagulative necrosis. So the coagulative necrosis, why it called as coagulative necrosis is nothing but here what happens is the structural proteins in the cell and also the enzymes get denatured. So there would be a uh, proteolysis of the dead cell or blocked because the enzymes which is used for the proteolysis also denatured. So what happened? There will be no proteolysis. So the necrotic cell would preserve or would remain for some time. Okay, they would not be suddenly phagocytosed. So that's why there would be an intensive eosinophilic appearance would be appeared for some days or weeks. Then after that they are broken down by the lysosomal enzyme from the leukocytes. So this coagulative, the area for the coagulative necrosis we will call as infarct. Like we will call it as kidney infarct, all these things, spleen infarct, all those things. Now liquefactive necrosis, they are opposite coagulative necrosis because there would be a digestion of the dead cells. So after the dead cell digests, they transform into a viscous liquid. So we call it liquefactive necrosis. And here the necrotic material would be creamy yellow because of the presence of leukocyte when we call it as pus. Then gangrenous, actually gangrenous is not a type of necrosis. For example, in gangrenous we have two wet gangrene, dry gangrene. Wet gangrene come under liquefactive and dry gangrene come under coagulative. Then caseous, it is most common, which is mainly observed in tuberculosis infection. Caseous necrosis is the main characteristic feature of tuberculosis infection. So here what happens is granuloma are seen. So here a small like space of granuloma is seen, and we will also see the surrounding leukocytes, and we also see the epithelioid cells, all these things. Here why it is called caseous means because it looks like cheese like appearance. Then fat necrosis is mainly the pancreas, acute pancreatitis. So here, there would be an damage. The fatty acids combined with the calcium to form and chalky white areas, that is fat saponification. So here in histology, this necrotic area appears shady outline of necrotic cells. Then fibrinonecrosis is nothing but it is a vascular damage. Okay, so it's a vascular damage, and here it is there would be a deposition of plasma proteins and immune complexes which leak out of the vessels. So there would be a bright pink amorphous appearance. That is what fibrinoid we call that fibrin like. Okay, so that way we call this necrosis fibrinoid necrosis. So that's all we have completed the necrosis. So by this, the part one of the pathology of the cell injuries completed. So let's see the apoptosis and the remaining topics in upcoming parts. Okay. So thank you for listening. Hope you understand. If you have any doubt, you can comment below. And also, if you want this notes in a PDF pattern, you can comment below. It will be available in description.